Chair, ladies and gentlemen, the etiology of abdominal aneurysms is known as thought. Indeed, the new mediated process is thought to be many contributing factors to this disease process, and this is due to the evidence in the current literature. In particular, is the presence of chronic inflammatory cancer within the aortic wall. It's thought that immune cells infiltrate the aortic wall where there is abdominal tube tissue and tube media. And it's here that it's thought it causes degradation of the cell matrix, collagen and elastin fiber disruption, and more importantly, smooth cell apoptosis. And ultimately, this results in loss of structural integrity, and aortic wall weakness, and formation of aneurysm. And this is depicted in the figure here, where on the far right you see a cross-section of an aneurysm, where you have marked thinning of the tube immediately to the yellow section, and the formation of angiogenesis. It is clear that the critical event in the development of aneurysm disease is a smooth cell apoptosis. Well, these cells produce the structural components that are required for structural integrity. However, it's unclear how autologous immune cells cause death of autologous smooth cells in the aortic wall. Our group is interested in the natural killer cells, or NK cells, which are a small but highly cytotoxic component of the infiltrate. We've experienced with two findings: that there are high levels of these cells in patients with AAA compared to age-matched controls, and secondly, that NK cells isolated from patients with AAA displays increased cytotoxicity against smooth cells in in vitro clear assay. Hence, our hypothesis is that NK cell cytotoxicity contributes to the autologous smooth cell apoptosis that's observed in the aortic wall of AAAs. Now, NK cell cytotoxicity is a delicate balance between both activatory and receptor signaling, with inhibitory signaling predominating. Now, inhibitory signaling is lost in the absence of MHC class 1, which can include tumour transformation or viral infection of target cells, and this results in activation of one of the 4 key receptors. In particular, I'd like to draw attention to the latter, NKGC. Now, this is important because the ligands for the receptor are well known and well characterized, unlike that of the other 3, which are unknown. And most importantly, these ligands are expressed in tissues that are affected in autoimmune disease, such as rheumatoid arthritis, multiple sclerosis, even in the aorta of tachycardic arthritis, and in experimental autoimmune diabetes. And it's based on this, it was our aim to investigate whether or not a particular phenotype characterised NK cells in AAAs. We recruited 2 groups of patients, those with abdominal aortic aneurysm and age-matched controls. We initially isolated peripheral blood cells in both patient groups using multi-parametric flow cytometry that looks at 4 key activating receptors. However, we were able to demonstrate the difference between the percentage of NK cells expressed either 4 receptors, or indeed the cell surface density, which was measured by measuring the mean fluorescence intensity. As we previously demonstrated that NK cells in patients with AAA have increased cytotoxicity, we attempted to see if we could out potential difference by activating these NK cells in 2 patient groups. What we find by activating them with the classical NK cell activator IL-2, that patients with AAA, or NK cells in patients with AAA, displays increased levels of C69, which is an early activation marker, which is associated with availability, and this may be available in vivo. As the site or the tissue injuries to the abdominal aorta, we went on to investigate and read specific phenotypes to these NK cells. So at the time of vascular repair, we sampled intraluminal blood. And again, we looked at the activator receptor phenotype, but again, we were able to demonstrate no obvious difference between intraluminal blood or peripheral blood using flow cytometry. We then went on to isolate tissue NK cells, and although we had a sample of 10, we were only able to sufficiently isolate NK cells from small samples due to the inherent difficulty of getting sufficient much for analysis. And although on the whole there was no difference between the 2 areas, either peripheral blood or the aortic wall, what was interesting is that the cell surface density of NKGC on tissue NK cells, which is enlarged here, was significantly reduced. And what this suggests is that within the aortic wall, there may be potential ligands for this receptor that results in natural receptor downmodulation. To this effect, we then went on to isolate smooth cells from sections of aneurysm tissue taken time of open surgery. And this slide shows you the smooth cells in culture which show the characteristic Killing Valley effect. To ensure that these were indeed smooth cells, we characterised them with indirect immunofluorescence with 2 smooth muscle markers, and over 99% of cells that we isolated were indeed vascular smooth muscle cells. And we found, using flow cytometry, that from all samples, smooth muscle cells express ligands for NKG2D, in particular those from micro A and micro B. We then went on to try and corroborate the findings that we found in the tissue by culturing NK cells with the aortic smooth cells that we explanted from sectional findings in the tissue. And again, we found that you have a steps-down modulation, like we've shown with the tissue NK cells. So, in conclusion, Mr. Chairman, ladies and gentlemen, there's no characteristic NK cell phenotype that finds AAAs. However, we've demonstrated that NK cells in patients with aneurysms have a lower threshold for activation, and this may relate to their cytotoxicity. Tissue NK cells have a reduced surface density of the receptor NKG2D, which suggests they may be ligated to the ligand from the aortic wall. And indeed, we've demonstrated that smooth muscle cells isolated from the aneurysmal wall express the ligand for NKG2D. We propose a potential model whereby NK cells infiltrate the aortic wall, where they interact with smooth muscle cells via NKG2 and the common ligands. This results in aberrant activation, despite normal levels of MAC class 1, resulting in degranulation against smooth cells and ultimately smooth cell apoptosis. The findings that we've demonstrated here today have also been corroborated by a group from the States who looked at aneurysmal tissue using gene expression profiling, and they found that the genes for the NK cell killing pathway were significantly upregulated compared to control aorta. So clearly, the NK cell killing pathway is an important immune-mediated process in aneurysms. I'd thank my supervisors, surgeons and leads, and for the institution for supporting my research. Thank you very much.
Questions or comments?
I've got a problem with your control tissue. Was it normal aorta that you were using, or was it atheromatous aorta? Because an abdominal aortic aneurysm also has atheromatous cluster within its wall.
We didn't. The problem was that all the tissue that we isolated was from the second time open surgery for aneurysms. We didn't have the control aorta to look at with regards to the expression of these ligands. That's something that we need to do. But the problem is just trying to get.
If it was in your control tissue, I must be going balmy.
No, we just got aortic tissue from the time of open surgery for AAA only.
Right. The second thing is, are you hypothesising that there's a whole genotype of people who are more likely to get it because they've got a reduction of this ligand?
We're actually saying that these ligands are potentially upregulated within the aortic wall, and this results in inappropriate activation of the immune cells, in particular NK cells, and this results in autologous death of smooth cells.
Very nice. Have you looked at any other immune response other than NK cells?
No, our group is specifically focused on the innate system, but many of the investigators looked at both CD4 positive T cell and macrophage, and for instance, one group has shown that macrophages kill the immune cell by fast ligand interactions, and it's thought that CD4 positive T cells promote inflammation in the aortic wall by secretion of particular interleukins. But we concentrate on NK cells.
Question in front?
No, previously, that's
I'm delighted to see embryology moving from cancer into vascular disease. It really is a new point. But more seriously, can you do anything to address the cause and effect question?
No, that's a valid point, and that's something that many investigators are trying to establish whether or not the information in the aortic wall is actually the cause or indeed the effect. And obviously, the problem with aneurysm and the work that we did at the end stage is getting experimental models have shown that the inhibiting information in the last stage of calcium chloride is that inhibiting information did inhibit the formation of an aneurysm in the mouse model. But of course, obviously, in humans, because we're dealing at the end stage, it's difficult at the moment to establish what those truth or again.
Okay, thank you very much.